Okay, so I'm Eugene Lin, or you can call me Frank. Okay, I am one of the 6 teachers in charge of these courses. So these 2 weeks, I will introduce 3 chapters, chapter 10 and 11, biomembrane structure and the transmembrane transport of ion and small molecules. Next week, I will introduce chapter 20, integrating cells into tissues. Okay, so after my part, the Professor Liao will deliver 2 more courses, and then it comes with the first exam. Okay, I can tell you my exam all come from what I teach in the course. Okay, will not come from anything I didn't mention about. Okay, so just be noticed they will be the first exam on March 20th. Okay, we didn't ask you to buy the textbook, but our contents all come from mainly come based on these 3 textbooks: the Molecular Cell Biology by Harvey Lodish, and the Molecular Biology of the Cell by Bruce Elbers, and the Cell Biology by Jurek Karp. Okay, in case you need to read more detail. So to help you to prepare the content after course, I prepare my slide like this. It looks really busy, but it can help you to understand what I teach and what's the key concept in these slides. I write down the key concept and description about the slide in red, and also point out some supplementary information of the figures in blue. For example, I write it down, this protein, there will be a lot of polar ring here and non-polar tunnel here. Okay, just put some key point to describe the important information about these figures. Okay, if you have any question, just interrupt me and raise your hand. Okay. So because this is the first course of the advanced cell biology, I would like to discuss this: why cell biology is so important. Kind of this question. Okay. So we are in the College of Life Science and Medicine. Okay, so every faculty or researcher focus on different topic. For example, immunology, neuroscience, cell biology, tumor, anything else, development, okay, evolution. Okay, so we study different topic. But what's the most important question, or what's the most fundamental question? Do you have any ideas? Everyone study the same goal. They have the same goal. Okay, so the same goal, the fundamental question in biology of life science, to me, is what is life? How does life work? Okay, what kind of stuff build up life? Basically, we are kind of complex of the protein, right? Or plus some DNA, nucleic acid, or some ion. But why we think we are alive? But protein we will not consider them alive, right? So which component build up the life? What's the life? How does life work? That's the most fundamental question in biology. And if you check these slides, this is the slide to describe the living organism come from the really simple molecule like atoms. Then atom can build up the molecules. A molecule can assemble to the macromolecule like DNA. And different DNA can form the next level will be the organelle. And cell contain different organelle. Different cell can build up the tissues. Different tissue can work together to form the organs, and the organ can form the organ systems, then finally build up the organism. Okay, so there's a huge gap right here. Okay, above this slide, everything are not alive. They are dead stuff. Okay, but below this line, all of this we can consider they are alive. They are living things. So cell is the simplest or the best unit of the life. So to me, cell is the kind of perfect model organism for us to understand what is life, how does life work. Okay, because inside the cell, everything are not alive. How can we control a dead stuff? We need to use, we cannot, they are not, we cannot communicate with them. Okay, so only thing we can control them is based on the basic physics or chemical principle. Okay, actually the cell also control all of that stuff based on the physics and the chemical, basic physical and chemical principles. So if you know the detailed mechanism, you will know they all come from the really basic, like they can use hydrophobic, they can use charge, they can use different such a property to build up the really complicated living environment. Okay, that's I would, if you learn the cell biology, I would like to recommend you to learn based on this concept, how those are different stuff they can run together based on the physics and the chemical principle, and finally build a really complicated living environment or living reactions. Okay, so just I think this video come from the YouTube is really good. So then just take a look. Okay.
Every living thing on this planet is made of cells. Basically, a cell is a protein-based robot too small to feel or experience anything. It has the properties we just assigned to life. It has a wall that separates it from the surroundings, creating order. It regulates itself and maintains a constant state. It eats stuff to stay alive. It grows and develops. It reacts to the environment, and it's subject to evolution. And it makes more of itself. But of all the stuff that makes up a cell, no part is alive. Stuff reacts chemically with other stuff, forming reactions that start other reactions, which start other reactions. In a single cell, every second, several million chemical reactions take place, forming a complex orchestra. A cell can build several thousand types of protein, some very simple, some complex micromachines. Imagine driving a car at 100 kilometers an hour while constantly rebuilding every single part of it with stuff you collect from the street. That is what cells do. But no part of the cell is alive. Everything is dead matter moved by the laws of the universe.
Yeah, moved by the law of the universe. Okay, so that's what I recommend you to kind of try to learn the cell biology based on this concept. Okay, any question so far? No? Okay, so let us jump to the chapter 10, biomembrane structure. I will also teach by this way. In this chapter, it's all about hydrophobicity. Okay, I can tell you, which is a kind of physical property, right? So how to use hydrophobicity to kind of introduce this biomembrane structure, okay? So as I mentioned before, the cell is the fundamental unit of the life, okay? To build up such a life environment and then perform the life reaction inside the cell needs to uptake nutrients and the essential molecule. After those reaction, physiological reaction, they need to release the waste or kind of move out such exchange molecules, okay? And all this molecule exchange happens to the plasma membrane. What you have learned, this is lipid bilayer structures. It's our plasma membrane, okay? So the physical or chemical property of those biomembrane is basically they have a hydrophobic core or hydrophobic layer, shown by these yellow colors. So after this class, you will know how the molecule transport within and across this membrane. They need to deal with this hydrophobicity. And how do protein locate on the membrane? They also need to deal with this hydrophobicity, okay? So this is a cartoon to show the component of our biomembranes. So the cell membrane is composed of different lipids and proteins. So the lipids showing in the gray colors, and the protein, there are multiple proteins showing by the different colors. The major lipids are phospholipid, okay, the lipid conjugate with some phosphate group. And depends on the way that protein incorporate into the membranes. Membrane protein can be classified to three groups: the periphery membrane protein, and a periphery membrane protein, and the integrate membrane protein, and the lipid anchor protein that we will introduce next chapter. Okay, so we'll introduce lipid first, then introduce the protein in the next chapter. So this is the still image. It didn't tell you, let you know how those molecules behave. Okay, so to describe how they behave, in the 1972, two researchers, Single and Nicholson, they collect the research from different scientists, and propose one model to describe how those membrane molecules behave. Okay, they propose a fluid mosaic model to describe how they behave. So they think that those lipid bilayer behave like some respect like a two-dimensional fluid. They only move in the two-dimensional sheet. Okay, and in which a lipid and a protein molecule can diffuse easily. Okay, as you can see here, those are lipids. Okay, and if you see the side view, you will see they move easily through in such a two-dimensional sheet. Okay, they can easily. We call this electro diffuse. Okay, and those are lipid or protein. They are not evenly distributed on the membrane. They actually, some of them can form the cluster. Okay, so for example, this we call the lipid raft because there are a lot of lipid and a certain specific protein will accumulate there. So we call this lipid raft because it's like the raft in the river. Okay, the lipid-based raft. So we call that lipid raft. So if you take a look for those biomembrane, you will see there are some cluster in the membrane. So we call it's display the mosaic pattern. That is why we call this fluid mosaic model. Okay. And then the textbook just give you some idea to describe what's the property of those membranes. The first, the membrane can bend to different shape. They can change the shape a lot. For example, this is the EN, electron microscopy graph, to show the surface of a human cell that has been infected by HIV virus. So this cell has been infected by HIV virus. Then the HIV will kind of resemble by themselves and build up the new virus particle on the infected cell. Okay, so those are nascent HIV virus particle. They try to budding out from the membrane. So they will use the membrane from the host to build up their shell. Okay, and try to budding out. So the membrane can bend like this. And in many organelles, this is Golgi apparatus. Okay, the cell is all covered by the membrane. So we have a different cisternae like a cis, middle, trans. So they can also show the kind of build up those membrane-bound structure by different shape. And actually, those membrane is super dynamic. They can be super dynamic. Those are a live cell image I will show you. The eagle view of the living cells. Those cells try to migrate to their destination. And as you can see here, they keep showing such a wave-like pattern right? They keep changing and bending their membrane. We call this a membrane roughly. Okay? So those membrane roughly is really important for cell to extend the cell mass and for them to migrate. Okay? Yeah. So they can be really dynamic. Okay. And in the different cell type, they can change their shape a lot. For example, this red blood cell is a disc-like shape. It's all covered by the membranes. And this is the multicilia. Multicilia is the kind of hair-like structure on the surface of the cell. Okay, so in our airway, epithelial cell in our airway, there are a lot of those airway cells. They have a lot of multicilia on the cell surface. So those multicilia can bend it and generate the float. Okay, to help our airway to remove the mucus. Okay, and reduce the inflammation. Okay, so those cilia, if you're under the animal, you will see they are keep bending. Okay, so all of this cell extension all covered by the membrane. No matter what they extend by such a stick out pattern, they all covered by the membrane. Okay, and even this sperm have this tail called the flagella. It's all covered by the membranes, and this is multicilia, and this is a primicilia. Okay, multicilia can be only found by certain type of the cell, like airway or some organs. But the primicilia can be found in 94% in cell in our body. And it is normally only one cilia can be found per cell. One cell have one cilia. Okay, they are really tiny on the cell surface. So in this case, you just have one primary cilia. They cannot move. They basically serve as a cell antenna to receive the chemical or mechanical signal from the extracellular space. Okay, to antenna to sense anything happens from the environment. Okay, so if you're interested why this cannot move, why this can move, probably we can. Those cilia structure, they are inside the cilia, they are microtubule, one kind of cytoskeleton. Okay, so if you enlarge those microtubule pattern, multicilia, there's you probably have learned this before. Multicilia, this is microtubule. This is a section view. If I take a section view from this cilia, you will see 9+2 pattern. You probably, this is a one microtubule filament, okay? Microtubule filament. So there will be kind of 9+2. So 9 will be 1, 2, 3, 4, 5, 9 on the outside here, okay? But there's two more microtubule rings in the centers, okay? So those are multicilia. And why they can bend? Because there are some motor protein connect here and connect here. There are some motor protein connect here and connect here. Okay, and they try to kind of move the microtubule filament like this. So that is why your cilia can start to bend. Okay, but primary cilia is a 9+0. They don't have such a central two microtubule filaments. So they cannot move. They don't have motor protein to help them to move the two kind of microtubule filament. Okay, so that's before people think this is a non-functional structure, but finally people think there are a lot of receptors on the surface of those primary cilia. They are really important. If these things, if defect happens to the primary cilia, you will have big trouble. You will kind of have organ abnormality, obesity. You cannot see, you cannot hear, you cannot smell. Basically, it's just disaster. Okay, yeah, that is why this is so important. Why I introduce so many? Because I studied primary cilia. Okay, yeah. Okay, so all of this extending structure covered by the membrane. Okay, and it can keep everything inside the cell will not leak out. Okay. Okay, so next we just take a look of the really quickly review the major component of those biomembrane. As I mentioned before, is a phospholipid. Okay, so first you need to learn this term, amphipathic. So what is amphipathic? Amphipathic is mean the molecule contains two parts with very different chemical properties. Okay, so we would say phospholipid is amphipathic because it contains a hydrophilic polar head and hydrophobic fatty acid tail. Okay, usually we draw this cartoon. This is a polar head, and it's a chemical structure like this. It's a hydrophilic. It's come from my microphone. It's It's a hydrophilic head. Okay. So annoying. And this is this two tail is a fatty acid chain. It's hydrophobic. Okay. So you need to learn the most important thing you need to learn is hydrophobic molecule aligned with hydrophobic molecule. Hydrophilic aligned with hydrophilic molecules. Okay. There is the energetic favorable condition. So if you mix different phospholipids in the environment, they will try to align with each other. So that is why the fatty acid tail will align with other molecules, fatty acid tail. And the polar head will align with other polar head, okay, based on those chemical properties. So that is why if you put the lipid in the water or aqueous environment, those will be the hydrophilic environment. This side will be also hydrophilic environment, right? So they will hide those hydrophobic fatty acid tail in the middle, and try to explore their hydrophilic polar head to the two side, face to the hydrophilic environment. So that is why you will see the plasma membrane or cell membrane is lipid bilayer. If you use this as the EM, electron microscopy graph to show the membrane structure. Okay, so osmi is a heavy metal which can strongly bind to the polar head group, this side, these guys, okay? And that is why you will see the distance, there's a gap between these two polar head. And you can easily calculate the thickness of the lipid bilayer. Usually it will be the 4 nanometer, 4 to 5 nanometer, okay? And this is a cartoon to show you how the membrane basically build up. So in the beginning, they may align with each other and form the two-dimensional sheet, but those white colors show the hydrophobic layer, okay, which were exposed to the hydrophilic environment, which is not energetically favorable condition, right? So they will try to hide those hydrophobic layer. The way they do this is form the bow, okay? And align their hydrophobic layer together. And finally, they will form, if they are enough lipid, they can form the two-layer lipid bilayer. We call this liposome. This is a section of those bow. If there's less, lipid can only offer this micelle is one-layer lipid structures. Okay, so just to let you know, a lot of liposome or micelle, which they are really popular recently because have you heard about the mRNA vaccine? They need to those can serve as a vehicle to deliver mRNA and can do the mRNA vaccine and cure a lot of diseases. Okay, but before you deliver those mRNA, you need some vehicles. Okay, that would be these guys. Okay, I think the people build up those vehicles also got the Nobel Prize. Yeah. Okay, so it's a basic condition. The liposome, because it have a hydrophilic environment here and hydrophobic here, so you can mix, basically you just mix those lipid with your chemical or with your antibody. They will build up this liposome with your drug, with your component you try to deliver, okay? So they will incorporate the hydrophobic molecules will incorporate it here, and the hydrophilic molecule will, for example, DNA is hydrophilic, so you bind either here or here, okay? And the hydrophobic drug will be incorporated in the middle. Then this can be serve as a vehicle to deliver those stuff in your body, okay? Okay, so there's three major classes of the lipid: the phosphoglyceride, sphingolipid, and steroid. We will introduce them one by one. So phosphoglyceride will be the major, the basic structure is they will be polar head and two fatty acid tail. And depends on the polar head, they have been named as a PE, PC, PS, and PI. Okay, so PE stands for the phosphatidylethanol-amine, and the PC stand for the phosphatidylcholine, and PS is phosphatidylserine. PI is a phosphatidylinositol. Okay, so depends on the head. The PE and the PC don't have charge, but the PS and PI are negative charge. Okay, and in some case, there's also other type of the phosphoglyceride is a plasmalogen. Okay, because they link their polar head with a different linkage is ester linkage. Those are PE, PC, PS, PI, they link by the ester linkage. Okay, ester linkage is more stable. It's more stable than this ester linkage. Okay, and the sphingolipid will be right here. Okay, the second class. The sphingolipid look really similar to the phospholipid, but they are not fatty acid tail. They are sphingosine tail. Okay. They also have a two leg, but it will be the sphingo leg, okay? So those are all sphingolipid come from the sphingo, that is why they belong to this sphingolipid. And such a sphingomyelin is the most abundant sphingolipid. As I mentioned, the structure and the shape of the sphingomyelin is really similar to that of the phosphoglyceride. And also some a polar head of those sphingosine could be the kind of sugar. Okay, for example, this 6-carbon sugar can serve as a their polar head and conjugate with those sphingosine. We call this a glycolipid or glucosyl-ceramide. Okay, it would be this one conjugate with a sphingosine. So glycolipid contains 2 to 10% of the total lipid in the plasma membrane. So it's also the kind of major lipid you can find. Find in on the cell membrane. Okay, then will be the sterol. So those are 3 kinds of sterol: cholesterol, ergosterol, and the stigmasterol. So the basic structure of the sterol is a 4-ring, as you can see here. There's a 4 yellow ring base hydrocarbons. They are hydrophobic. As you can see here, this OH group is a polar group. It's combined to the water. And this part is a non-polar hydrocarbon tail. And those are 4-ring base hydrocarbon are also hydrophobic. So they will have this structure. Sterol have a huge hydrophobic, relative huge hydrophobic part, and really small the polar group. Okay, so cholesterol is abundant in the plasma membrane in the mammalian cells or vertebrate cells. Okay, but you cannot find the cholesterol in bacteria like a prokaryotic cell or plant cell. Okay, so in the plant cell, they have a similar structure called a stigmasterol, and some bacteria or prokaryotic cell will be the ergosterol. Okay, cholesterol can be also found in the vertebrate or mammalian cells. Okay, and as I mentioned before, the hydrophobic site is too big. The hydrophilic site relative is too small, so they cannot kind of build up the lipid bilayer on. Okay, so they need to kind of they only can kind of incorporate to the phospholipid bilayer, but they still, as you can see here, the hydrophilic part allied to the polar head, and the hydrophobic part allied to the fatty acid tail. Okay, but they cannot form a lipid bilayer by themselves. If you just purify the cholesterol and mix them together, they cannot form such a 2-dimensional sheet. Okay, they need to mix with the phospholipid and form those 2-dimensional sheet. So those sterol, they kind of can align here and generate some space. And then to prevent those lipid to package or to packing. Okay, to compact. Okay, to generate some fluidity. So the fluidity is important. If the membrane have too bad fluidity, everything cannot move. Okay, and the molecule protein cannot move. Everything just stick there, they cannot move. And the molecule cannot exchange. They cannot go in and go out, they cannot cross the membrane. If the membrane fluidity is too high, everything will get lost and the structure cannot maintain the integrity and everything just disrupt. Okay? So you need to keep the good, proper fluidity for the membrane. So what kind of factor determine the membrane fluidity? First is the lipid composition. So these are the cation of the unsaturated lipid. These are the saturated lipid. Saturated lipids have a really rigid and straight fatty acid. Okay, so if you put the saturated lipid, they can really align with each other and form a really compact complex. They don't have any space to move around. So in this case, this will be really viscous and really fluidity is really low. Okay, anything just stay there, they cannot move. But if you put some unsaturated lipid, their fatty acid tail have a kind of some part of them will have a double bond. All of this, the saturated lipid, they all come from the single bond, chemical bond. This part of them will be double bond, and then can kind of generate such a kink or twist. Okay, so they have such a kink fatty acid tail and can kick others, their neighbors away from them and generate some space. In this case, they have a better fluidity. Okay, so unsaturated fatty acid have better fluidity. Saturated fatty acid have a lower fluidity. And the temperature is also the major determining factor. Higher heating, like if you put the oil in a bowl, heat those oil can become the solution, right? So have a better fluidity. But if you put them to the freezer, the lower temperature will freeze them and they become the gel-like pattern and reduce the fluidity. So temperature would be the also major determining factor. But as I mentioned before, the proper fluidity is really important for the animal or for us to live, right? We need to kind of have proper fluidity. That is why we can exchange molecules and keep our membrane structure intact, okay? So, but some animal, they need to live, like some bacteria or some fish, they need to live in a really extreme environment, like a super hot and a super cold environment. How can they maintain their membrane fluidity to the physiological condition? They need to have they need help with, for example, need help from the cholesterol. With the cholesterol, they can narrow down the range even in such a high temperature environment. They still can keep them with a proper fluidity range. Or they can help the animal to adapt to the really cold environment, okay, if your lipid have cholesterol, okay? And as I mentioned before, those lipids, they can easily lateral diffuse in such a two-dimensional sheet, but they cannot flip without any energy, okay? So as this cartoon, those lipids, you can see this chemically, if you use a chemically principle to define this structure, there will be the hydrophobic layer and the two hydrophilic layer here. So if you don't, against this chemical property like this, lateral diffusion will not always follow this hydrophobic and hydrophilic. But if you want to move this upside down, move this lipid upside down, we call this flipping, okay? This polar head needs to, against the hydrophobic layer, then move to the other side. You need energy to do that, okay? Without energy, that is why you can only do the lateral diffusion, okay? And basically, those are lipids without any other hinders or any block event. Naturally, chemically, one molecule, their lateral diffusion rate can be the 1 micrometer per second, okay? So the mammalian cell, usually the diameter would be kind of 10 to 20 micrometer. So basically, if one molecule, they want to move from the one side to the other side, it's only take 20 seconds, which could be really fast, okay? So that is why I want to let you know the lipids are really dynamic. That is why, because those lipids, they exchange a lot, okay? Yeah. So how to measure this lateral diffusion or the movement of membrane molecules? The most popular assay we call the fluorescence recovery after photobleaching, or FRAP, okay? FRAP. So those can measure the lateral movement of the protein or lipid within the plasma membrane. So basically, first you need to label your component of interest. For example, you need to label the membrane protein, you need to label your membrane lipid by certain fluorescent dye. Okay, this is based on fluorescence microscopy assay. So this is cartoon. You basically, if you label the phospholipid, you will see the whole cell just really bright. You don't see the dot, okay, because you don't have enough resolution to see the single molecule. Only probably some method can do that, like a single molecular image, but the general microscopy don't have a resolution to see each single molecule. So you just see the whole of those whole cell are bright, okay? You don't see each single lipid. The way how to define their movement, you need to kind of use a strong laser and shine the strong laser to the region of your interest. For example, would be right here. You want to see how fast this labeling molecule moving in these regions. You just shine the laser here. So the fluorescence have one chemical property. You just keep shining, the laser will make their fluorescence signal become dimmer. They become dark, okay? But the molecule is still there. They're still moving, but their fluorescence become dimmer, okay? But it only affect the molecule here. The molecule, fluorescent molecule outside of your region of interest, they still can be visible. Only this part, because they're receiving your laser and receiving the reaction called photobleaching. That is why they become dimmer and they cannot be detectable anymore. But those stuff still can go out, and other things still can go in. You just cannot see those are dark molecules. So after a while, you can imagine those labeled molecule, unaffected molecules still can gradually go into these regions. And their diffusion rate is totally depends on how fast they can kind of recover or refill this region, right? Okay, if they move really fast, they take less time to refill this region. If they don't move at all, they will not refill your bleaching area, right? So if you measure, just use some software to draw the circle here. You will see this is x-axis is the experimental time. So in the beginning, you will see it's a really bright here. That's a basal level of the fluorescence intensity in this region. And then you shine the laser. Usually it will take maybe 5 to 10 seconds to shine pulse laser, okay, to achieve the photo bleaching. So the fluorescence, the y-axis is the fluorescence intensity of those molecules in this region. It will decrease to make sure the bleaching happens. Then we turn off the laser, keep monitor what happened, how long those green molecule will go back to this environment. So they will gradually go back, and to the saturated level, okay? So you can get the two information from the flap. The first is how fast of this molecule go back. So you can easily calculate the slope, okay? And then know the, for example, the half time and how long it takes to reach this saturated pattern. And the next is mobile fraction. It's this mobile fraction, okay? In this case, that's a mobile, it can only recover to the 50% of the original level. It means it's only have a 50% mobile fraction. 50% of half of them are immobile. It means if even those stuff not those are green dot, only half of those green dot they are moving. The rest of them don't move at all. Okay, that is why it only covered half of the original label. You get my point? Okay, so we can get the mobile fraction and the recovery time. Okay, so just take a practice. So those ER endoplasmic reticulums and this nuclear envelope. So a researcher just shined the laser on these tiny rectangles. It bleach, and you can see the recovery, right? Let me play again. So which one you think the mobility will be faster? This one or this one? Can you tell? This one? Raise your hand. This one move, ER molecule move faster or nuclear envelope move faster? This is 1, 2. Okay, 1, 2. Can I tell? Can you tell? You don't, it's too bright here. Can you tell? So basically the ER membrane, after bleaching, they can gradually recover, right? But the nuclear envelope, it still keep it in dark. So basically the nuclear envelope stuff, they don't move at all. That is why they cannot recover back to the bleaching area. But the ER, they can gradually move back to that bleaching environment, okay? That is why we think ER mobility, electron diffusion rate will be faster than the nuclear envelope, okay? And this is the Golgi apparatus. You can also kind of bleach this area, and then it will gradually go back here, okay? So those will be the flap. If you don't understand, just you can ask me later, okay? Okay, so the cell membrane, as I mentioned before, the cell membrane is a lipid bilayer. The two layer composition are different. So we call this cell membrane is asymmetric, okay? If they have the same composition, we will call it they are symmetric. So they are different, so we call this asymmetric. Okay, so and there will be two major type of the lipid bilayer, shown in the orange and the gray colors. So in the orange colors, you can find this on the membranes and on the surface of the organelle. Those are orange leaf left all face to the cytosol. Okay, they share the same composition. But the other one would be the exoplasmic phase lipid raft, lipid layer. Okay, they showed in the gray color. They will face to the extracellular space or face to the lumen of the organelle. Okay, they have the same compositions. So those are asymmetric or orientation determined by the ER. Okay, so basically most of the lipids synthesized in the ER. So the ER, they will synthesize lipid and they will arrange what kind of lipid will go to this gray layer or go to the orange layer. And after that, those vesicle or even gauge actually come from the ER. Gauge generate from the ER, okay? So those ER will keep generate the new vesicle and then will share, those orientation will be conserved and move out, budding out until the plasma membrane. So if I make it simpler, the ER will generate those vesicle and those orientation will kind of keep all this orientation. The gray will face gray left, lipid left will face to the lumen, and then but fused with the plasma membrane, it face to the extracellular space. The orange one will always face to the cytosol. Okay. Yeah, and the membrane protein, most of the membrane protein, I would say maybe all of the membrane protein are synthesized in the ER. Their orientation kind of, for example, this end will face to the cytosol, this end will face to the lumen, are all determined during their synthesis. Okay, I guess Professor Liao will introduce this part later. Okay, so and this orientation will keep conserved as well. Okay, yeah. After fused with the plasma membrane, this will face to the extracellular space, and this end will face to the cytosol. So in this great lipid lift, it's sphingomyelin, phosphatidylcholine are bound in the exoplasmic leaf left. Okay, and this is a less fluid. Okay, but in this orange lipid lift, it's a phosphatidyl in ethanol, amine, and phosphatidylserine, phosphatidylinositol are bound in the cytosolic leaf left. Okay, and the cholesterol distribute evenly in both these two lipid bilayer. Okay. And this table in the textbook, it's kind of a little bit boring. It just showed you, it measured the lipid composition from the different sample. It sometimes come from the human neuron, sometimes come from the bacteria, plant. As you can see here, there's some key information I summarized here. For example, cholesterol doesn't exist in most of the prokaryotic cells, so you will only find cholesterol in the human or but not to those plant or to those bacteria. Okay, will be 0 here. And phospholipids, sphingolipids are synthesized in the ER and the Golgi. So that is why you will see if you collect those lipids from the Golgi or ER, they will be abundant. Those lipids will abundant to where they synthesize, which makes sense, right? Okay, that's it. Okay, it's a kind of really boring table. Okay, and the lipid composition can also determine the shape of your membrane. For example, the PC, if this lipid bilayer come from assembled by the PC, the thickness could be 3.5 nanometers. If the PC aligned with the cholesterol, they can further extend their fatty acid tail, which can reach to the 4 nanometers. But the sphingomyelin, it can reach the 4.6 to 5.6 nanometer. Even the cholesterol aligned with those sphingomyelin, they don't further expand those thickness of the membrane. Okay, and the PC is chemically is a colon column-like shape, and the P is a cone-like shape. So in this shape of the membrane, you will see more PE here. It will change. We call this a membrane curvature. Okay, yeah. So lipid composition can also change the shape of the membrane. And as I mentioned before, it's a fluid mosaic model, right? The mosaic, the term is come from the factor that those are lipid or protein, they are not evenly distributed on the membrane. Some of them can form a cluster. For example, the lipid raft. So in the lipid raft, there are a lot of certain signaling molecules, or for example, cholesterol, sphingolipid, and some specific protein will form a cluster and form such a lipid raft. Okay, they have a less fluidity in the lipid raft and the better fluidity outside the lipid raft. Okay, why they need such a lipid raft structure is because to generate or to turn on certain signaling, it all depends on the concentration. You need to kind of locally achieve certain amount of the concentration of the signaling molecule. You can, or over is a threshold, then you can activate that signaling. Okay, so lipid raft will be the factor or the mechanism of cell used to generate enough amount of the signaling molecule locally. Okay, then that is why a lot of signaling molecule will accumulate here. They can, because there are more or enough receptor concentrate here, they have ability to receive the signaling or ligand from the extracellular space, then trigger the downstream molecule. Okay, if only one receptor, they cannot do anything. Okay, so you probably, have you heard about the LLPS, lipid phase separation? Liquid phase separation. It's kind of, cell can also generate some oil drop like a pattern or just bubble inside cell. Okay, they can also locally enhance the protein's concentration locally and then turn on some signaling. Yeah, that's really emerging field and a really hard field. Yeah. Okay. Okay, so those is one kind of cell label. So chlorotacin B is the mark, and this GN1 is the marker of the lipid raft. So we'll see they look like this is the surface of cell and this is the DNA. They are not evenly distributed on the membrane. They accumulate here. Okay, it'll be really enriched. They kind of serve as a signaling hub here. Okay, and generate some downstream signaling. Okay, and if, as I mentioned before, the ER is a major place to organelle to synthesize synthesize lipid. If they synthesize too much lipid, they can form the kind of they will accumulate cholesterol or triglyceride in the space between the two lipid leaf lap. Okay, lipid bilayer here. Then finally form such a micelle, only one layer of lipid cover, and we kind of put the lipid to the core of this micelle. We call this a lipid droplet. Okay, so those lipid droplets are also synthesized from the ER. If you eat too many McDonald's, I would not say McDonald's, Western food, or to eat too many fat, cholesterol, okay, your cell will accumulate, will generate more triglyceride and the cholesterol. Then you will see your cell will form more lipid droplet. And you can use some lipophilic dye to label those lipid droplets, like oil red. Okay. Okay, so just quickly summarize these sections. The first, to describe how those lipid molecules behave and the membrane protein behavior, we call this a fluid mosaic model because they can really electrodiffuse in this two-dimensional sheet, and they are not evenly distributed in the membrane. They sometimes form a cluster, so that is why they can display the mosaic patterns. So that is why it's called the fluid mosaic models. And the second, you will see those are membrane. They are hydrophilic polar head on the two sides, and there's also a hydrophobic layer in the middle. Okay, and those phospholipid, they are amphipathic, which contain hydrophilic and hydrophobic part. And the lipid, there are three major lipid: phosphoglyceride, sphingolipid, and steroid. And the membrane, they are asymmetric. So PC, SN, they are bounded in the exoplasmic lipid left. Okay, PE, PS, and PI, they are more abundant in this lipid left. Okay, and the cholesterol are evenly distributed to the both lipid bilayers. And lipid composition can affect the thickness and the shape of the membranes, and lipid protein that can laterally diffuse within the membrane, and their dynamic can be measured by the FRAP, fluorescence recovery after photobleaching. Okay, and I also introduced the lipid RAF, which is a cholesterol, sphingolipid, and certain protein-rich microdomains, which can kind of really important to trigger some signaling. Any questions so far?